Thank you. Thanks very much to the organizers from SOFIN and to Jeff Abbott for the invitation to speak here. It's a really special treat to have a meeting like this come to you and have this opportunity to present in front of an audience that includes a lot of collaborators, mentors, as well as lab mates. Thank you. So we heard in the last couple of days talks from Adam Paraputian about the discovery of the piezo channels and the important roles that they play in physiological processes. So my lab studies piezo-1 from a molecular to a cellular to an organismal scale. And today I'm going to share with you some of the things we've learned about the role of piezo-1 in physiological processes and how these insights instructed our development of a new tool to study it. So I'll start with a project led by Jamie Nurse, project scientist in the lab, that looked at the role of piezo-1 in brain development. Now, one may not think of the brain as a mechanically dynamic organ, but during the development, the cells in the brain encountered a lot of different mechanical cues. These include the hydrostatic pressure in the fluid in the ventricles of the brain as it's forming. This fluid is beaten back and forth by cilia lining the apical region, which generates a shear flow. And then there is also tissue stiffness, which changes as the organ grows. In addition, there are cytoskeletal forces that are produced by the cells that cause movement and connections between the cells. So we wondered whether piezo-1 might be involved in regulating brain development, given that there are so many mechanical cues. And so we used a mouse model, a constitutive mouse made by Adam Paraputian's lab, and looked at the brains in the wild-type mice and in legitimate knockout embryos. So this is at mid-gestation at E10.5, and we noticed that the entire brain seemed quite underdeveloped. The neuroepithelium was much thinner, smaller, and when we zoomed in, we saw that the knockout brains are smaller. They're also disorganized, as particularly disruptions at both the basal as well as the apical border. We then looked at differentiation. So at this stage, you have mostly neural stem cells. Neuronal differentiation has begun. And when we stained for a neuronal marker TUJ1, we saw that the knockout brains had much less, had fewer TUJ1 positive cells, suggesting that differentiation was also compromised in addition to proliferation. So this is in vivo. We also harvested neural stem cells from these mice, and we studied them in vitro and recapitulated both the proliferation as well as the differentiation defects, indicating that these effects are cell autonomous. So to understand how these phenotypes may be arising, we dissected out the brains from wild-type and knockout mice and did bulk RNA-seq analysis from them. And we were really astonished to find that the top pathways to come up were related to cholesterol biosynthesis. And here I'm showing you the cholesterol biosynthesis pathway. And each red dot represents an enzyme, the gene for which is downregulated by at least 1.5-fold. So 16 enzymes in the pathway, there's about 70% of the enzymes are downregulated by at 1.5-fold, and a few more that don't quite meet that stringent threshold. So based on this, we wondered whether cholesterol levels might be reduced. We used Philippine, which is a fluorescent probe that binds to free cholesterol, and we stained the neural stem cells cultured in vitro, and we saw that the knockout neural stem cells had lower cholesterol levels. So then we asked whether we could use cholesterol supplementation to rescue the defect. So we differentiated neural stem cells in vitro. Here I'm showing you neuronal differentiation. Knockouts, as you can see, have much less neuron formation. But then when we supplemented it with cholesterol, we found that we could rescue the neuronal differentiation and get more neurons forming. And we could do the same thing also with astrocytes, where cholesterol supplementation increased the amount of astrocyte formation. So to summarize this part of the story, we see that the absence of piezo-1 drastically impacts neural development, the brain development, and that the cholesterol biosynthesis pathway is affected, and it underlies at least some of the phenotypes that we observe in the knockout mouse. Okay, so this was at the tissue scale, but to really understand how piezo1 functions, we need to understand how it responds to mechanical forces. And after Adam's lab identified the channel in 2010, a number of labs showed that it responds to a lot of different types of mechanical stimuli that you can impart onto a cell, such as poking the cell, stretching its membrane, squirting fluid on it, and so on. But cells also produce mechanical forces. Just like you or I, given an object, would poke it or push it or pull on it to determine whether it's soft or stiff, cells are constantly interrogating whether their environment is soft or stiff, and they use that information in order to make decisions. And they interrogate substrate stiffness by making additions to the substrate through focal additions, and then using actomyosin forces, they burn ATP to generate pulling forces that are imparted onto the substrate. And the cell is basically asking, does the substrate give? If it doesn't, it pulls even harder, generating a larger traction force. So we wondered whether these cell-generated traction forces, which are orders of magnitude smaller than the mechanical cues that we can impart onto the cells, whether they might activate the piezo channels. These forces are typically in the single digits or piconewtons in amplitude. So to study this, we had to move away from, we had to rethink of the standard assays for piezo1 activity, which has, of course, been patch clamp electrophysiology, because the process of patch clamping, it disrupts the cell-generated forces itself. And so we came up with a non-invasive way of studying piezo1 activity using the fact that the channel passes calcium, and so we can use fluorescent calcium indicators. And then by doing the imaging in turf microscopy, which looks selectively at the cell-substrate interface, we can look at the channel activity where traction forces are being communicated to the substrate. So we did a very simple experiment where we loaded cells with a calcium indicator, such as fluor4 or cal520, and then we imaged in turf microscopy. And when we processed the data, we found that there was a lot of calcium dynamics in the wild-type cells, but in cells where we had knocked out piezo1 using CRISPR engineering, there was a drastic reduction in the signals. And these signals appear as transient flickers. We call them piezo1 calcium flickers. So around this time, I started collaborating with Ian Parker's lab, and what has been a really long and fun collaboration. Ian's graduate student, Kyle, had developed a computational algorithm for taking signals such as these and making high-resolution cellular maps, super-resolution cellular maps of the signal. So you can take a video of piezo1 calcium flickers like this, and Kyle's algorithm, Flicker, returns a map of active piezo1 channels. And we saw that the activity, so every region where you see a flash of light appears as a white dot here. The activity is not randomly distributed all over the cell. It appears in hotspots. And so now the question became, do these hotspots of piezo1 correspond to regions of high traction forces? So I collaborated with Alex Dunn's lab at Stanford University to use a FRET-based membrane tension probe that they had generated. So we combined our approach to make piezo1 activity maps of cells together with their approach to do traction force measurements and make traction force maps of cells. So this is data from the traction force sensor, and here blue indicates regions of high traction forces. So now when we overlay the two maps, we see that there's a very high correspondence between regions where there are traction forces and where piezo1 is active. So in other experiments that I'm not going to show you the data for, we also pharmacologically either increased traction forces or decreased it, and saw a corresponding change in the piezo1 calcium flickers. So essentially, we found a new modality of activating piezo1 channels by piconewton scale cellular traction forces. And this happens in a spatially regulated fashion in that a piezo1 channel that happens to be close to the force-bearing addition experiences the force and activates, whereas the channel that is farther away remains silent. So we were, of course, now curious as to the physiological roles of this cell-generated piezo1 activity. And so over the last few years, we found in work that I did as a postdoc with Francesco Tombola and Lisa Flanagan that it regulates stem cell differentiation in neural stem cells. With Wendy Liu, we found that it influences the macrophage inflammatory response to stiff implants, such as surgical implants that generate an inflammatory response of the macrophages. And in collaboration with Hardem Paraputian's lab, we found that it influences cell migration during skin wound healing. In other work that we did with Mike Cahillan's lab, we also saw that piezo-1 influences the inflammatory response of regulatory T cells in a mouse model of multiple sclerosis. But in this case, we don't yet know fully what kind of forces is contributing to the piezo-1's role here. Okay, so around this time, we were also beginning to get interested in where piezo-1 is located. So to look at the location of endogenous piezo-1 channels, we used a mouse made by Hardem Paraputian's lab where the endogenous channel is tagged with a TD tomato fluorescent reporter. So we took these mice, harvested cells from them, and then imaged the TD tomato in turf microscopy. And we saw that the channel is distributed on the cell surface as these puncta. And we were really surprised when we imaged these cells over time to see that the puncta were mobile. And looking at it closely, some puncta moved quite a lot, and then some not so much. And there were regions of the cell that were reminiscent of focal adhesions, where the cells tend to get, where the channels tend to get enriched, and they don't seem to show a lot of mobility in those regions. So we are now interested in teasing out what relations there might be between the channel mobility and the activity. And we got some clues as to the importance of where the channel is over space and time in our studies on skin wound healing and keratinocyte migration. So when we get a skin wound, the keratinocytes at the edge of the wound migrate into the wound bed to reinstate the epithelial barrier. And this process is known to be mechanically regulated, but the mechanotransducers involved in it were not well understood. And so we wondered whether piezo-1 might be involved. Now, Hardem's lab had made conditional piezo-1 knockouts in the skin, and they did wound healing studies where they made full thickness skin wounds on the dorsal skin of mice, and they looked at the wound size 6 days after wounding. So in knockout mice, they found that the wound sizes were smaller, that is, wounds were healing faster, whereas in gain-of-function mice, they found that wound sizes were larger, that is, wounds were healing slower in these mice. And so in my lab, we wondered how this might be happening, and so we did a lot of live cell imaging to try to figure it out. So one of the first experiments we did was to harvest keratinocytes from those mice and image their migration in vitro. And so here is a wild-type control cell, and this one is a piezo-1 knockout. And the piezo knockout is going way faster, which you can also see in the mean square displacement plot over here. So this is in single cells, but wound healing happens with collective migration. So we used an in vitro scratch assay, wherein you make a monolayer you culture a monolayer of keratinocytes, and then you make a scratch, and you image the process of the cells migrating to close the wound gap. So I'm showing you here control on the left and knockout on the right. And again, the knockout cells are migrating faster and closing that wound gap faster. We did this in gain-of-function cells as well, and saw that those were slower. And also in the case of just the control cells treated with URA1, a chemical activator of piezo-1, we saw that the effect was really striking. I mean, in the URA1 treated cells, the monolayer barely advances at all because of it's such a strong effect. So we wanted to know where is piezo-1 and how is it doing this? And so we, again, used the piezo-1 TD tomato mice, harvested keratinocytes from them, and looked at where in the cell the piezo1 channels are located. And so this is, again, our punctate distribution of the channel. This is done in turf microscopy also. But then when we saw in our sample cells that were migrating, the distribution was different, was polarized and more enriched at the back of the cell. So on the back of the cell is where the maximal retraction forces take place as the cell is pulling its tail to detach and be able to move forward, so to speak. And so this gave us clues as that piezo1 might be involved in the retraction. So this is in single cells. We looked again now in monolayers. So again, we did an in vitro scratch assay, but now instead of using a 10x objective, we used a 60x objective. And in addition to doing DIC imaging to look at where the cells are located, we also did turf imaging. And Jesse Holt, the graduate student in the lab that did these experiments, gets great credit for making these are really difficult experiments where we are doing turf imaging for 16 hours as the monolayer is migrating. And so we saw that the piezo1 localization is quite dynamic, even in these monolayers. And this is, we are looking at a small region here. And occasionally, there are regions where piezo1 gets enriched at the wound edge. So let's play that movie again. So you can see the monolayer advancing there, channels moving around, and then we're going to see an enrichment form just around here in a moment. There we go. You get a band-like structure, which then dissolves. Right? So we stared at these videos for a long time, and then a pattern began to emerge. So not everywhere in the wound edge, at the wound edge, do we see enrichment of the channel. So in regions that don't show enrichment, the wound edge processively moves forward. But then in other regions where you do see enrichment, that enrichment is followed by a pulling back. So instead of going forward to close the wound gap, the cells are migrating and moving backwards. And so we think that this enrichment is eliciting retraction locally. And that is why when you have an absence of piezo1, you get faster wound healing because you don't get these backward motions. But then when you have situations with increased piezo1 activity, you get slower wound healing. So this work suggested to us that to understand piezo1's functions, we shouldn't be thinking about just when is it active, but also where is it during as the biology is happening. And so it's important to track both activity as well as location of the channel. But current tools don't allow that. You have to pick either one or the other. And so we need new tools to study this. And so we've been working to fill this gap. And we took iPSCs and knocked into the piezo1 locus a halo tag domain. So wherever the channel is produced, wherever the channel protein is, it's actually tagged. So we did this on both copies. So all channels are tagged with the halo tag domain. Now the halo tag is a bacteria. We also checked that attaching the halo tag domain didn't disrupt the activity by doing patch clamp electrophysiology. I'm not going to show you the data, but okay. So the halo tag is a modified bacterial enzyme that has the ability to covalently label itself when a chloroalkane ligand is presented to it. Now, this ligand can be functionalized with a number of different things, such as fluorophores, biotin, cross-linkers, et cetera, which makes it a really versatile system. And we made this, we did the CRISPR engineering in human-induced pluripotent stem cells, so HIPSCs, which can be differentiated into many different types of cells. So when we differentiated the cells, we can then label it with a fluorescent halo tag ligand. And so this is a cell that's been labeled. And just like we did with the TD tomato, we can image these over time. And you can see the motion of the puncta. And you can see occasionally multiple puncta come together into a spot like over here. Now, the greater brightness and photostability of these new JF dye-based halo tag ligands allow us to follow individual puncta with greater precision and for longer periods of time than we could do with the TD tomato reporter. the fluorophore can also be switched out. So now in the next experiment, instead of using a ligand that is just a fluorophore that's always glowing, we used a calcium-sensitive ligand, so it glows brighter in the presence of calcium. And now I'm showing you here a cell that's been labeled with the BAPTA probe. And we don't see as many bright puncta as we did in the earlier experiment because not all the channels are active, and so you only see the active channels, the other ones are dimmer. We can follow an individual punctum over time. We can image its brightness over time. And we see that it starts off dim, and then there are these brief periods of it getting bright, which we attribute to the channel opening, calcium coming in. The halo tag, the calcium-sensitive halo tag ligand is just sitting right below the pore, and so it binds to that and increases its brightness, this reporting on the activity. So to confirm that, we treated the cells with Yoda-1, and immediately it became obvious that we can see a lot more puncta, a lot more of the channels are active, so you see a big difference in the number of bright puncta. And when you follow an individual punctum over time, you again see that it's spending more time in the bright state, which would be consistent with a channel that's been treated with an activator that encourages it to be open more. So here are the traces that I just showed you, but now put next to each other. We can, with this trace, we can make an all-points histogram, just like we do with patch clamp traces, and we see there's a big peak corresponding to the dim, which we attribute to the closed state, and smaller peaks corresponding to the bright state. And in the presence of Yoda-1, you get an increase in the amplitude of the bright peaks at the expense of the dim peak. Right? We also see multiple open levels here, which we interpret as there being multiple channels that can activate in a diffraction-limited spot, and we are now doing more quantitative experiments to confirm this interpretation. Okay, so we've made, with this halo tag approach, we can now study the activity of the channels at temporal resolution that begins to approach that of patch clamp electrophysiology, but in cells that are intact, that haven't been poked with an electrode, and in a format that allows us to monitor the activity of multiple piezo-1 puncta at the same time. And in a given field of view, we can get dozens to hundreds of channels at a time. And so we can begin to look at where in the cell are channels active over time. An advantage of doing this genetic engineering in iPSCs is that we can differentiate them not just into any type of cells, but also into tissue organoids, which recapitulate human physiology. And so we've taken the first steps in the organoid front with neural organoids. So here I'm showing you 2.5-dimensional neural organoids called micropatterned neural rosettes. These are models of the neural tube. You can, using bioengineering approach, you can make them very reproducibly so that they have a single central lumen, and then the neural stem cells organized radially around it. So we made these organoids from our piezo-1 halo tag iPSCs, and we can label them with fluorescent halo tag ligand and look at the distribution of the channel in this organoid. So we see an enrichment of the channel at cell-cell borders and also at the apical region. So this is just one image. So this is a confocal slice. We also did this using light sheet microscopy. And when we quantify the piezo-1 puncta location over many samples, we see that they are enriched at the lumen. So this is close to the lumen, this is far from the lumen, and so they're enriched in those regions. So here is a light sheet video that we took in collaboration with Gokul Upadhyayula at UC Berkeley. This is on Eric Betzig's mosaic light sheet microscope. It's a live neural organoid. In white, you're seeing a live actin stain. So this is rotating it on the side, and in blue, Briefly, we're going to see nuclei, just to give you a sense of the architecture of where the cells are located. Now we zoom in to the actin stain, to the actin image. We can make our, so we've labeled with our piezo-1 halo tag ligand, just the plain fluorescent one. And we see, in addition to the small puncta, we also see these ugly autofluorescent blobs. We know these are autofluorescence, not real signal, because they're also there in the piezo-1 knockout organoids that we imaged. But because they have a very characteristic appearance that's different from the puncta, we can computationally separate them, filter out the autofluorescent blobs, and look just at the piezo-1 puncta, which are shown as here in magenta. We've segmented out the piezo-1 puncta and shown them in magenta, and we're just slicing through the organoid now. So this is just a proof of principle that we can image, we have the ability to image piezo-1 at a molecular scale in tissue organoids. And we're really excited about all of the studies this opens up. So to summarize, we generated a new tool that allows us to look at the localization as well as activity, the spatial temporal dynamics of both, of endogenous human piezo-1 in native cellular condition at single channel resolution in a variety of cell types, because the iPSCs can be differentiated into a number of different cell types as well as tissue organoids. And we can, of course, also use the iPSCs for doing human disease modeling. And a lot of mutations in piezo-1 are now emerging and being associated with human diseases. So this really allows us, this opens up a number of clinical translational studies as well, where we can try to figure out the biophysics underlying the diseases. So we're really excited about using this tool to do a number of new studies, and look forward, once our validation is complete, to also share this. I think this will be an interesting tool for the whole community. And I would like to end with thanking an incredible group of collaborators as well as funding agencies, but most of all my lab, which has been just amazingly fun to work with, and I can't wait to see what we do next. Thank you very much for your attention, and I'm happy to take questions now.